We performed an exploratory proteomics discovery. It's an unbiased approach. This methodology we were able to detect around 2,000 proteins in the cerebrospinal fluid of patients in the trial at baseline and end of study. So we can look at the change from baseline in these different biomarkers detected by this discovery proteomics method, and look at treatment effects. So key biomarkers are presented at this meeting that we identified to be pharmacodynamic biomarkers of CT1812 or zervimesine. And we also identified correlates to the favorable decrease in that neurodegeneration biomarker I told you about, neurofilament light or NFL. We've identified other correlates to that might reflect biomarkers of neurodegeneration, and of course the favorable decrease in that with the zervimesine. So that was really exciting. And then lastly, as I mentioned before, ADAS-Cog11 was our primary efficacy outcome measure for the trial, where we saw favorable effects with zervimesine, and we identified molecular correlates to that favorable effect with ADAS-Cog11 with zervimesine. So, this is building out our biomarker program and also enabling us to understand what the biological impact is of zervimesine on patients with Alzheimer's. So really exciting.
We've obviously talked a lot about efficacy, but can you just give a little bit of overview on what the safety of the drug looks like, what we've seen across this study and just the exploratory studies as well?
Yeah, absolutely. So zervimesine exhibits a very nice safety and tolerability profile to this point. That we, in our 6-month trials, we had a trial Spark earlier on with mild to moderate Alzheimer's patients that was really read out at 6 months, but we had some patient data at 12 months. And overall, the safety tolerability is very good. Now in the SHINE trial, getting into more specifics, there were adverse events at the 300 milligram dose of zervimesine, but at the 100 milligram dose, there were no adverse events at all. So that's very promising, and we believe from our, looking at our trials that we've done to date and all the safety data as a culmination, that it appears we've identified a dose range for phase 3 that has efficacy, a good efficacy signal with a very low, no troublesome adverse events.
Yeah. Oh,
so
that's encouraging to say the least. Yeah.
Absolutely, and that absolutely helps us with our phase 3 planning to identify the right doses or dose that we would use.